Welcome back to my channel. இன்னைக்கு இந்த வீடியோல நம்ம வந்து ornithine cycle அதாவது யூரியா cycle பத்தி பார்க்கலாம். How the urine is formed in the human being. Mechanism of urine formation in human. அதுதான் வந்து நம்ம இப்போ பார்க்க போறோம். ஸோ இதுல வந்து இந்த சைக்கிள் பார்த்தீங்கன்னா உங்களுக்கு தெரியும். லிவர்ல வந்து நம்மளோட இந்த நைட்ரஜனஸ் வேஸ்ட் ஃபார்ம் ஆன நைட்ரஜனஸ் வேஸ்ட் வந்து நம்மளோட பாடியில் ஃபார்ம் ஆயிருக்கும் இல்லையா, ஆஃப்டர் த டைஜஷன் எவ்ரிதிங். ஸோ அது வந்து எப்படி வந்து பிரேக் டவுன் இன்டு யூரியா ஆகுது, எப்படி இந்த யூரியா சைக்கிள் லிவர்ல வந்து நடக்குது, அதோட டீடைல்ஸ் தான் இப்போ நம்ம பார்க்க போறோம். The nitrogenous waste which is formed as a result of the breakdown of the amino acid is converted into urea in the liver by the cycle called as urea cycle or ornithine cycle. So glutamine அப்படின்ற அந்த அமினோ ஆசிட்ல இருந்து நமக்கு வந்து ஃபர்ஸ்ட் ஸ்பிளிட் ஆகி வர்றது வந்து NH4. ஸோ இப்போ NH4 என்ன ஆகும் அப்படின்னா, NH4 வந்து ஃபர்தர் கன்வெர்டட் இன்டு கார்போமைல் பாஸ்பேட் பை த ஆக்ஷன் ஆஃப் என்சைம் கார்போமைல் பாஸ்பேட் சிந்தேஸ் 1. அதே போல் டைரக்டாக குளூட்டமைன் வந்து கார்போமைல் பாஸ்பேட் சிந்தேஸ் 3ன்ற என்சைம் மூலிமா டைரக்டாக கார்போமைல் பாஸ்பேட்டாகவும் நமக்கு இங்கே கன்வெர்ட் ஆகும். தென் இட் இஸ் ஃபைனலி கன்வெர்டட் இன்டு சிட்ருலின். ஸோ தென் சிட்ருலின் வில் கன்வெர்ட் இன்டு arginine succinate, then into fumarate, then into arginine. So from the arginine it will enter to the cell cytoplasm and that is converted into urea. அப்போ remaining ornithine வந்து again it will get into the mitochondria of the liver cell. So இது எல்லாமே எங்கே நடக்குதுன்னா mitochondria of liver cell. arginineல இருந்து மட்டும் நமக்கு வந்து cell cytoplasmக்கு மூவ் ஆகி அது ஃபர்தராக வந்து யூரியாவை கன்வெர்ட் ஆகும். ஸோ இந்த யூரியா again drained out into the blood and that is excreted in the form of urine by the filtration occurs in the kidney. So this is how the urea cycle takes place inside the liver. So urine formation happens in 3 main processes. So that process is, first one is glomerular filtration, second one is tubular reabsorption, and third one is tubular secretion. As I said before, the urine formation is formed in 3 steps. First one is the glomerular filtration. Glomerular filtration is nothing but the filtration happening inside the glomerulus. So the blood will enter into the kidney first, then it will enters into the renal artery. That means blood enters the kidney from the renal artery. So blood is actually in the renal artery that enters the kidney now, and then what happens? See that it enters into the glomerulus. So this is how the glomerulus gets the blood. So what happens after that? See that the blood actually is composed of what all it contains. It contains water, and it contains colloidal proteins. It contains sugars, it contains salt, and along with that the nitrogenous waste. So all is carried by the renal artery into the glomerulus. So now we will see the first step now. What is the first step? Urine formation. For the urine formation, the first step is the filtration of the blood that should take place in the glomerulus. This filtration we call it as glomerular filtration, which is a passive process, not active. It is a passive process. That is the filtration of the glomerulus. Glomerular filtration is a passive process. The fluid that leaves the glomerular capillaries and then enters the Bowman's capsule. So what is the Bowman's capsule? This is the Bowman's capsule, right? So this is the Bowman's capsule. So here what happens? See that actually the glomerular filtration is a passive process, and that the fluid will leave the glomerular capillary and enter the Bowman capsule. So this filtrate only we call it as glomerular filtrate, which enters the Bowman's capsule from the glomerulus. So the fluid which is entering, that one we call it as the glomerular filtrate. So the glomerular membrane, now we'll see about the glomerular membrane. The glomerular membrane is the one which has a large surface area and is more permeable to water. So the glomerular membrane is permeable to water and small molecules present in the blood plasma. Then the blood enters the glomerular faster with a great force through the afferent arteriole. What is the afferent arteriole? The entering arteriole into the glomerular. So while it is entering, it will enter with a greater force. While it is leaving, it will leave slower with the afferent arteriole. So in afferent arteriole, which is carrying the blood to the glomerulus, is faster. When it is leaving the glomerular, it is slower. The arteriole which leaves the glomerular is termed as efferent arteriole. Fine? Okay. So this is the two arterioles which is present inside the glomerulus. Okay. So the force is because of the differences in the size between the afferent and the efferent arteriole. You all know if the size is very bigger, it will flow freely, right? If it is smaller, it will flow slower. So like that only the afferent arteriole is wider, so it have a great force, but the efferent arteriole is narrow then compared with the afferent arteriole. So the force differs between the two arterioles. So the glomerular hydrostatic pressure created here is nearly about 55 mmHg inside the glomerular. Okay. Then the kidney have to produce about 180 liter of glomerular filtrate in 24 hours. So the molecules such as water, glucose, amino acids, nitrogenous substances, so they all will freely flow from the blood into the glomerular. Okay. So the molecules which is larger than 5 nm, the size, so that will be barred to enter into the tubule. That means what? They won't enter into the tubule in the filtrate. So in glomerular filtrate, the molecules which are larger than 5 nm will not enter at all. So the glomerular pressure is the actual chief force which is pushing the water and the solutes out of the blood and across the filtration membrane. So the glomerular blood pressure, which is approximately 55 mmHg, is much higher in the other capillary beds. What are the other capillary beds? In the renal tubular, around the renal tubular, there are lots of peritubular capillary beds, no? So that one only they are saying here. The two opposing forces are contributed by the plasma protein in the capillaries. Okay, so mostly the osmotic pressure is 30. The osmotic pressure is 30 mmHg, and the capsular hydrostatic pressure is 15 mmHg. So see the difference. Colloidal osmotic pressure is 30 and capsular hydrostatic pressure is 15 due to the fluids which is present in the glomerular capsule. So the net filtration is about 10 mmHg, which is responsible for the renal filtration. So we will see it now like this, how the calculation is done here. So what is the net filtration? So the net filtration, how we will calculate? We need to add glomerular hydrostatic pressure, and from that we need to minus colloidal pressure plus capsular pressure. So colloidal and capsular we all know 30 and 15. So what will be the filtration rate now? We all know 55 mm Hg is our glomerular hydrostatic pressure. From this we need to minus the colloidal osmotic pressure 30 plus the capsular hydrostatic pressure 15. So the answer is 10 mmHg. So this is the pressure which is present in the net filtration, which is the net filtration pressure. Okay. Okay, now coming to what is mean by ultrafiltration. So the 10 mmHg which results in the ultrafiltration is nothing but the glomerular pressure. So the glomerular filtration rate is the volume of filtrate per minute in all the nephrons. So for example, the GFR, the glomerular filtration rate for the adult. For the adult, it is about 125 ml per minute. Per minute, in an adult, the glomerular filtration rate is about 125 ml. So 125 ml of blood per minute is filtered in the glomerular filtrate. Okay. So the blood from the glomerulus is passed out through the efferent arteriole. So in efferent arteriole, around it there is a muscle that is nothing but the smooth muscle which are able to contract, resulting in the vasoconstriction. So what is mean by vasoconstriction? Then you can answer this. What is vasoconstriction? See this table which is shown here. This shows the relative concentration of substances in the blood plasma and the glomerular filtrate. The glomerular filtrate is similar to the blood plasma, except if you see, there is no plasma proteins. See that in glomerular filter there is no plasma protein. So it is reabsorbed in the blood. So what it means? In cortical nephron, blood from efferent arteriole flows into the peritubular capillary bed and enters the venous system. So it will carry with the, so in the cortical nephron, the blood from the efferent arteriole flows into the peritubular capillary beds. So, and it will finally enters into the venous system, carrying with the recovered solutes water from the interstitial fluid that surrounds the tubule. So you can see this in this given table, that the proteins are just recovered in the blood, only the amino acid, glucose, urea, uric acid, creatinine, inorganic ions like sodium, potassium, chlorine, all these are as such enters into the glomerular filtrate. So from this what it means, the water and the protein, you can see that water is also entering, but the protein is not entering. So the complete protein is not at all entering into the glomerular filtration. It is reabsorbed and enters into the efferent arteriole, and then it is supplied again to the body. I hope you have understood this what I have said. The next one is the tubular reabsorption topic that includes proximal converter tubule, descending limb of Henle's loop, ascending limb of Henle's loop, distal converter tubule, collecting duct, and that includes tubular secretion also, which we will see in the next video. Thank you all.